If you look at your schedule, I'm talking about scaling genetic returner results, but it turns out that's what I'm talking about tomorrow. There was a bit of a program switch, so today I'm talking about some work that we've done in high-grade serous ovarian cancer, and this was originally the 45-minute talk for that section, so we're going to skip some slides that are in here. I've been pruning some slides in the back. So I'll talk about some things that if you're interested in single-cell sequencing and you're interested in connecting that to bulk tumors, that you may, in fact, want to know about. So first, we work on high-grade serous ovarian cancer. It's the most common form of ovarian cancer. It's not a diagnosis that's good to have. Clinical signs are usually, the disease is usually diagnosed pretty late because the early clinical signs are relatively common. And it's often already metastasized. And the final point that you need to know, there are transcriptomic subtypes that have been reported, and those subtypes are associated with survival. And so what we're going to try to do is figure out what the biologic basis of those subtypes is. And it turns out this is harder than one would expect. This is a quote that I love because it's attributable to Mark Twain, but in fact, there is no evidence that it's actually from Mark Twain, which is great when it's a quote about what you don't know. It ain't what you don't know that gets you into trouble, it's what you know for sure that just ain't so. There's a TCGA paper now from about 15 years ago that clearly defines 4 subtypes of high-grade serous ovarian cancer. We were very excited about this, tried to do some work on it, failed to build on it. And the student who was doing this is Greg, who is now faculty. So I'll give you an idea. He did a postdoc, is now faculty, but I'll give you an idea of what Greg struggled with as a student. We went to the TCGA paper, and there's this beautiful figure in that paper. It's this one. That makes it very clear if I look at this, I say there are absolutely 4 subtypes of this disease, non-negotiable, right? Like you could not make a prettier picture than this for a disease. Unfortunately, if you're Greg and you read the paper really carefully, you realize that to make this type of pretty picture, it turns out you have to remove a bunch of samples, the confusing ones. And it turns out that if you don't remove the samples, life is a little bit more challenging. So Greg did some work many years ago. We published this in 2016. That's kind of like almost forensic bioinformatics work, where we went back and tried to replicate what TCGA had done. And we found that they had removed some sample. They had not removed samples that probably should have been removed. And that induced a subtype clustering that is probably, it included samples that weren't this disease alongside these samples, so it really kind of makes weird stuff happen in the clustering. And so we make the argument that while there are some subtypes, it's really hard to say exactly what they are, and they don't look that pretty from the figure that I showed you. So a few years ago, we wrote a grant, really simple grant, just to figure out what's happening here. Really trying to probe this using single-cell sequencing, right? Because based on Greg's work, we thought that there were tumor microenvironment differences. So if you want to probe the tumor microenvironment, you do single-cell sequencing. The experiment is easy. And so if you do this, you can ask what your power should be. And so when we did this, we realized that we were going to need on the order of 400 to 500 samples to really be able to say anything conclusively. For single-cell sequencing, this puts us in a budgetarily challenging position. But bulk sequencing, it's really easy. There's lots of data sets that are already this size, especially if you combine them together. So all we need to do then is do something very simple. Take a bunch of single cell profiles and then use those to identify cell types, and then use really straightforward methods that exist and have been published over the last 10 years to estimate cell type composition in bulk tissue, which is essentially tissue RNA in a blender from a human tissue. And then life is easy. So if this actually worked, the talk would end now. It turns out this doesn't actually work the way one might hope. Sorry, I stole Ariel's punchline. So Ariel Hippen was a PhD student in the group and sort of really wanted to understand if this stuff worked. We designed an experiment where we took a tumor, did what most people do, which is RNA deplete bulk RNA sequencing. So if you're saying like, I want a tumor and I want to know what the gene expression of that tumor is, this is what's happening. We then dissociated cells from that tumor, and then we did poly-A single-cell sequencing. This is what you would usually do if you wanted to single-cell sequence a tumor. And then when we do deconvolution, all you're doing is comparing the box on the top with the box on the right, and you're basically saying, okay, I'm going to say in this bulk tumor, these are the proportions of cell types that are present using the profiles that I found in my single-cell sequencing. We also wanted to figure out if we could save some money, so we evaluated whether you can pool samples. So we pooled samples by basically combining different tumors together in the same reaction. There are two different ways we could do this, so we did both. So one is to do genetic demultiplexing, which means take naturally occurring genetic variants in the sample and use that to separate which cells come from which person. The second way to do it is to basically put an antibody on those cells with a barcode on it. So we did both of these things. There's actually a third way, which is to do barcoded lipids, but we had some technical challenges with the barcoded lipids. So we did barcoded antibodies and the genetic demultiplexing. But we also wanted to know if this actually worked in terms of gene expression values. So we also designed the experiment to test dissociation. So we ran rRNA deplete bulk RNA sequencing of the dissociated cells. So we dissociated the cells and then did bulk RNA sequencing using rRNA depletion. And we wanted to test the mRNA enrichment methodology differences by doing poly-A selection, bulk RNA-seq. So we have now bulk RNA-seq these samples 3 times and done 2 different single-cell sequencing runs on each one. So it gives us the ability to start to ask questions about whether the methodologies that get used actually work. And the tumors that we used here came from the OCRC Biotissue Trust at Penn. So first we can think about pooling. I anticipated when I wrote this talk that this was going to be 45 minutes, so we're going to blaze through this because this actually works. So there's 2 different methods, the hash demultiplexing and the genetic demultiplexing. At the end of the day, they largely agree, so that's good. So even though these are cancer samples, the genetic demultiplexing works. It's actually more sensitive than the hash-based demultiplexing. It has another benefit. Let's see if I put this slide in here. I did not put this slide in here because I just removed it while I was sitting in the back of the room. It turns out that the antibody-based demultiplexing appears to have maybe slightly different efficiency by cell type, which, if your goal is to understand the composition of a tissue, could be an issue. So we actually think the genetic demultiplexing is a better option in this type of setting than the antibody-based demultiplexing, which is great because it's also a lot cheaper, and it reduces the complexity of the single-cell run. So that was the simple part. Demultiplexing, yes. Dissociation. I am going to largely skip this part, but basically just say the dissociation looks real messy. There's stuff that comes up in dissociation that is clearly different between the cell suspension and the tumor. Things like, for ovarian cancer, the loss of adipose tissue, which we know and expect, but which makes a difference because many of the tumors metastasize to the omentum, and that can actually have survival implications. And so you may end up in a situation where outcome is confounded with the technical artifacts that your methods are introducing. So it is actually somewhat of an issue. And then mRNA enrichment. There are differences between mRNA enrichment. We think this is actually easier to deal with, but it's things like histones that, for example, aren't polyadenylated that can show up as kind of different between the 2 protocols. We think you could address this pretty easily just by filtering on a key set of transcripts. I will note that the mitochondrial RNAs are in this mix. So one of the things that people sometimes do is think about using mitochondrial reads to filter their single-cell sequencing data to say, okay, here's the amount of mitochondrial RNA I expect in my tissue. Okay, so I'm going to filter cells that have high mitochondrial read counts because that indicates a degraded cell. Makes a lot of sense. You should absolutely do that. On the other hand, if you are getting your mitochondrial estimates of your tissue from bulk RNA-seq, there are certain protocols, including an extremely widely used protocol, the Ribo0 Gold, that actually remove mitochondrial RNAs. And so you will dramatically underestimate the amount of mitochondrial RNA in your tissue, so you might set those thresholds way too stringently if you're not careful. Okay, and then deconvolution. So we end up looking at deconvolution from 2 perspectives. One is accuracy, which is how people usually do it. There's a set of methods that seem to work well. There's a set of methods that don't seem to work well. Again, I'm not going to dive into the exact details of which method is which, although I will say in our hands, Bayes Prism looked quite good on this. And then there's this other idea that we introduced, which is this idea of stability. So we make the argument that if you are going to evaluate deconvolution algorithms, one way to do that is to take a bunch of different experimental preps that represent exactly the same tissue, right? And if you do a bunch of different preps that represent the same tissue, what you want is a method that is accurate, so it seems to be giving you the right answer in simulation studies, because we actually don't know truth, we can just get close to it, and stable over a bunch of different experimental designs, right? Because the underlying biology is the same. It's only the experimental methodology that's different. So we should expect that method to be stable and accurate. And by both of these metrics, Bayes' prism was well-performing. So from here, I'll start to give like a little bit deeper dive into the results. So first, demultiplexing works, likely helpful. There are loss of certain cell types and enrichment of other cell types with dissociation. This probably is going to need new deconvolution methods. I'll get into this a little bit more in the second half of the talk. There are RNA isolation differences. I think these are possible to work around, but it's worth noting that the methods that currently exist and that are currently deployed in this space do not correct for this. So while I do think it is possible to do that, we are not currently doing with that in the literature that exists in the field, and there is a lot of it. And if you are interested in this, this came out last year in Genome Biology. There's a GitHub associated with it, so you can kind of say, well, look, you made a terrible assumption here. I'm going to make a different assumption. Let's see what the results look like. Okay, and then we did a paper where we applied the well-performing methods. I think I have hidden essentially every slide from this paper because of time. Basically, what we find is that if you're interested in high-grade serous ovarian cancer, there are recurrent patterns in the data. I probably wouldn't call them subtypes, but everyone else does, so rock and roll. They appear to be at least partially driven by the tumor microenvironment. And one of the things Ariel finds is that it looks like there are also differences in the cancer cell fraction, not just the tumor microenvironment, but it's very hard to untangle these. So if you're interested in ovarian cancer, I think this paper is a good jumping off point for a set of questions that are likely answerable with some kind of slightly new methods and slightly new experiments done. Okay. So then the thing I want to spend kind of a deeper dive on is some work by Adriana Ivic. So she wanted to ask this question, can you build a better deconvolution method with the information that actually exists in the data right now? We're not going to build that better deconvolution method. We just want to ask the question, is it possible? Is there evidence that the right information is present to be able to do this? There's a couple different ways you could do this. So first you have a tissue of unobserved cells that has some fraction of cell types, and each of those cell types has some gene expression profile. There's this idea I already talked about of doing bulk RNA sequencing, which contains cytoplasmic and nuclear RNA. One thing you can do is single cell RNA-seq. We've already talked about, you can lose cell types there. We think, for example, we lose adipose tissue. We think we get an overabundance of certain very round immune cell types. You can also do single nucleus RNA-seq. The benefit here is you don't lose the cell types. On the other hand, so this is nuclear RNA. So if we want to do deconvolution, right, we have a different set of RNAs present, but we're going to have all the cell types. So it's like I can have kind of very similar RNA profiles, but only from a limited set of cell types, or all the RNAs, but from all the RNAs, but different in terms of profile. So here's an example of a couple cell types that are lost. In this paper, on the left is single nucleus data, on the right is whole cell data. So you can, and they've been aligned so that the axes are consistent. So if you're going to argue with the UMAP, the only thing we're looking at here is the presence or absence of the same cell types in this UMAP. So I think this is a reasonable use of UMAP. I know that is up for debate sometimes. So one of the things you see is this like loss of adipose tissue here. The adipocytes are basically gone. And then also this loss of mesothelium. And this is well known. This isn't the only paper that points this out. But this is the type of thing that you see. And so you can imagine if you are using this sample on the right to do deconvolution of a bulk tissue, that sample is in fact not an accurate representation of that bulk tissue. And so that creates issues. People often still do this. And so Adriana's question was, well, if I do that, what happens? Like, is it bad? How bad is it? And if I look at the things that are not explained in that deconvolution, is there evidence there that maybe those residuals actually contain the information that I need? And so that's what we're going to do. We're going to estimate the proportions, and then we're going to use the cell type labels to compute some residuals. I'll show that in just a second. We've done this through a bunch of different approaches and methods. So the first one is we just use non-negative least squares. You might say, why would you use non-negative least squares? That seems like a very simple method that no one actually uses. Absolutely true, but we know these are compositional data, and if you imagine the absence of noise, non-negative least squares would be a reasonable approach. So we have that as kind of one of our control methods. And then we used methods that people actually use today, CyberSort and Bayes Prism, as well as a few different data sets, both data sets that are kind of very well characterized, this PBMC 3K data set that has probably been analyzed tens of thousands of times at this point for essentially the same type of thing, where we delete certain cell types. And then we also do data sets where we use proportions estimated from our single nucleus RNA sequencing data to mimic what would happen, estimated from our single cell RNA-seq data, but using our single nucleus RNA-seq data to give us a sort of more holistic representation of the tumor. And then we also do this with real bulks of high-grade serous ovarian cancer. I'm only going to tell you about one of these results just because of the limitations of time and wanting to have some opportunity to answer questions. So Adriana's hypothesis was essentially that the residuals associated with deconvolution, right, the unexplained expression, what you don't get when you multiply your cell proportions by your reference profiles, is going to contain information from the cell type that we are deleting. So if we take a cell type and we delete it, here we go. So we take our single cell RNA-seq, we have known proportions, we can make a pseudobulk from those proportions. We then remove a cell type, and that gives us a smaller single cell reference. And then we're going to do deconvolution of our pseudobulk, which had all the proportions, with this set with the reduced proportions. We can then take whatever these calculated proportions are, multiply them by our reference, and then take the pseudobulk, subtract this from the pseudobulk, and then get a residual. And then our idea is that if you do some factorization, like NMF, something simple, hopefully that residual factor looks like you're missing cell types. I'm only going to present one set of results, which is this realistic proportioned bulks with real missing cell types from adipose tissue, where we then add noise to the pseudobulks. So this is, it's actually this data set that I showed you before, which is why I chose to do this. And you can see we really lose the fat cells and the mesothelial cells. When we do this and we look in the residuals, we do see some evidence that we're able to, that the residuals from non-negative least squares, when you factorize them with non-negative matrix factorization, include factors that match both the adipose proportions and the mesothelial cell proportions. So we think the information's actually there, it's just in the residuals. So when you're applying the methods that you usually apply, you don't notice that this is missing, but you can actually recover this information, identify it's there. The same, I don't think I put this slide in. Yeah, I didn't put this slide in. So the same thing happens if you use CyberSort or if you use Bayes Prism, so the modern methods that people actually use. It's obviously clearer with non-negative least squares because that makes very strong assumptions about the data that we are violating. And then we're demonstrating that if we violate those assumptions, we can rediscover it. But the same thing happens in those more complex methods as well. So what this suggests to us, if we want to think about the takeaways for this, first, there are limitations of using RNA sequencing in this setting, particularly for complex tumors, where complex tissues like epithelial ovarian cancer, where there are a few other difficulties in this as well. So these are epithelial cancers, so you have some issues just getting your epithelial cells into single cell suspension and then running them through the microfluidic devices. But these limitations actually show up, and it's really important to know about them. There are a set of, for example, single cell profiles for this disease where the proportion of T cells seems, to me, unrealistically high. And one potential explanation for why the proportion of T cells is so high is that perhaps those are preferentially captured by single cell sequencing. And that's a guess as to what's happening here. We do think there are significant opportunities for new method development. So if you are looking for like, hey, what's my next paper? And you are interested in this topic, there's a few different methods that we've sort of put some guideposts on here. For example, you could imagine creating a deconvolution method where you run your deconvolution, but then you do some sort of factorization of the residuals and use those factors to then go back and try to deconvolve again. So you do some sort of iterative approach to deal with the lost cell types. You could imagine doing deconvolution from extremely large reference sets. So you could imagine, for example, taking the human cell atlas and using that as your benchmark for deconvolution. Or you could imagine kind of some hybrid of these approaches where you take the residuals, look for cell types in the human cell atlas, and do some deconvolution. So there are 3 potential methods papers right there. If you would like to publish any of those, we are not working on any of those, but I would love for someone to solve this problem in any of those 3 ways. So that's kind of opportunities for method development that exist. And if you look at our preprint, our real goal here is to, we're not setting out to produce a method, We are setting out to pose a question, and we think we posed the question reasonably well, and we hope that people will produce methods that will now answer the question so that we will not then have to do that work. So if you're interested, this is on bioRxiv. There's also a GitHub associated with it, so we thought that if people were planning to build methods, it'd be really nice to give them a starting point. So we think this can help you with your experimental design and assessments. Yeah. With that, hopefully I have 5 minutes for questions. Rock and roll. So I'd like to thank the folks in the lab who did work along these threads, so especially Greg, Ariel, Natalie, and Adriana. Collaborators, so the single cell work and a lot of our work in ovarian cancer is a collaboration with Jen Doherty at the University of Utah and Stephanie Hicks for the single cell work at Johns Hopkins, as well as their lab, as well as many other people, too many to list and have the names stand out. And then for the tumors from Penn that I talked about, those come from the OCRC Biotissue Trust. So Ronnie Dropkin, Dalia Amran, and Ihe Jung. And with that, I would be happy to take whatever questions you have. And if you are instead interested in biobanks, tomorrow afternoon I will talk about our biobank.